Let's look into some important factors which may be important while planning a genome assembly project. These may be number one is size of genome. Repeat content is very important because the assemblies they mess up if we have large number of repeats, and it's really hard to solve this repeat problem. If we have some related species genome assembled, that can help doing some kind of a guided assembly, and we can have advantage from this. And what strategy we are following, like what genome sequencer we are using, and what alignment, the analysis softwares we are using, that is also important. So let's look into size of the genome. Size of the genome can be estimated from the ploidy of the particular species in which we are interested. That ploidy means how many sets of the chromosomes are there. So individuals which are having like one set of chromosomes are they are called as haploid, and then normally the eukaryotes they are diploid. So for example, us we have two sets of chromosomes. Some species are triploids, and in this way this ploidy level can be up to many folds. So if we have those four sets of chromosomes, we call it as a tetraploid, and polyploid is the term if we have many sets of chromosomes. So that is an important component in genome assembly. How it will affect. So if we have the ploidy level is high, obviously we can be estimating that how many reads we should generate so that each position, each nucleotide position, and even their polymorphisms, we can capture that. So this thing is tackled in a terminology or in a thing which we call it as a coverage. So let's see what the coverage is. So typically we see for an assembly project, the coverage can be up to 100x. Let's see what is this. So coverage is the average number of times any given base in the genome is sequenced. So it can be derived by deriving the total length of acquired sequences by the genome length. So here we can have this equation where C is coverage, so it is NL divided by G, where N is the total number of reads, L is the length of the read, and G is the genome size. And the resultant answer what we are getting is represented as X. If its answer is 10, so it is 10X, so that is 10X coverage. So any nucleotide has been sequenced or visited 10 times. So here we try to understand this coverage by using this example. In this example, we see there is a Illumina sequencer that produced 1 million reads. Obviously, Illumina produces 100 base pairs. The read length is 100 base pair in Illumina. And for this species, the genome size is 1 MB, which is a million base pairs or million bases. So we use the equation C is equal to NL by G. So N is number of reads produced, which is this one, 1 million. And then into the read length, which is 100, and again the size is also 1 MB or million base pairs. So when we simplify that, we get 100x. So this genome has been covered as 100x. So each nucleotide is represented or is sequenced 100 times. Can we see it's an exact number? No, it's an approximation. It gives you a general or a rough idea about the coverage. So the coverage is not even as we see in this picture. So we have a genome and the reads are aligned onto that genome. So we see that some places in the genome got 5 reads, 3 reads, 2, none, 1 and 4. So this estimating the coverage by the previous equation is, as I said earlier, is kind of an approximate solution. But in real sense, this coverage is not even. And this uneven coverage has to pose challenges for our assembly process. So we see that effect of genome helps us in deciding which sequencing technology we can use and what computational resources will be needed. Obviously, we want to have more coverage if the genome size is big, the genome is complicated. So obviously, this is an important factor which we must consider. So genome size is an important factor in genome assembly, and this will help decide which sequencing technology we should use.